Hello lovelies, in this video we're going to be looking at classification and technology, all of the different bits that you need to know for your A-level biology.
Classification and taxonomy. Classification is the organisation of living things into multi-tiered or multi-level groups based on similarities in their characteristics. Organisms are classified into groups called taxons, and this system was developed by Carl Linnaeus. He developed it in around the 1700s, and it still uses Latin as its universal language, which can be the same around all of the world. This is helpful, so when you use a Latin name for an organism in every country, they'll know what organism you're talking about. We need to remember Carl Linnaeus and his name and how he developed this system. So when we classify organisms, what we do is we're putting them into groups. The reason classification is important and putting organisms into groups is important is because it helps scientists to know which species are which. So we're going to talk about domains in a minute as part of this classification, but Carl Linnaeus actually wasn't responsible for the domain. That comes later, and we'll look at that next. But I'm going to put it in so that you can get the full classification system as it is now. So he was responsible for kingdom onwards. It helps us to know which species are closely related to each other. It helps to identify organisms as separate species. So that's kind of knowing which species are which ones. So let's look at an example of a classification of a lion. Okay. So this is what we call the taxonomy of a lion. This is its classification. So these are the groups it is classified into. Okay, we need to learn the names of these groups or taxons in order. Okay, so domain, kingdom, phylum, class, order, family, genus, species. I remember this as what's for dinner, king prawn curry, or fat greasy sausages. Just to learn those first few, the letter of each one in the right order, because in the exam you'd be expected to be able to know those words in that order. You don't need to know these Latin words or names that are in each of the groups, okay? But they'll be given to you in the exam. But the actual words of the groupings, so the domain, kingdom, phylum, class, order, family, genus, species, you do need to know, okay? So this is the lion example. So it's a eukaryote, so it has a nucleus in its cell, so that's its domain. It's in the kingdom Animalia because it's an animal. It's in the phylum Chordata because it has a backbone, so a spinal cord. It's a mammal, so its class is Mammalia. It's a carnivore, which means it eats only meat, so it's in the Carnivora order. It's a big cat, and the big cat family is the Felidae family, so all the big cats are in that group. Its genus is Panthera, which is like panther, and then its species is Leo. So if we were to write this out as its binomial name, it's Panthera leo. Okay? So remember, binomial name is using the genus name and the species name. That gives it its official scientific name. So as we said, all these groups, all these taxons mean something. So every organism that has a nucleus in its cells that's made of eukaryotic cells will be in the eukaryota domain. Every animal, so every organism that's made of animal cells, will be in the Animalia domain. Everything that has a backbone or a spinal cord will be in the Chordata phylum. Everyone that's a mammal will be in the class mammals, so everything that has mammal features. Every organism that eats meat, roughly, will be in the Carnivora order, or that group. Every big cat is in the family Felidae, so the big cat features. And you can think of lots of big cats, jaguars, tigers, lions, cheetahs, all of those will be in that family. So something that shares all of these groups is really similar. They're really similar organisms. So all of those big cats are really similar to each other, okay, because they share lots of characteristics. That means they've been grouped in all these same groups going all the way down the taxons. So as we go down, we get more groups. So there's more families than there are orders. There's more orders than there are classes, etc. But each time there's going to be fewer organisms in each group until we get down to the species group, which is unique and identical. And there's only one single species, that is Panthera leo. Let's do a quick recap on what a species is, because we just talked about Panthera leo being the species of lion. And here we have Panthera tigris, which is the species name for tigers. So what do we actually mean when we say a species is this unique group where only one type of organism is in it? So the definition of species comes sort of in two parts. It's a group of similar organisms or organisms that share characteristics is kind of the first part, and that this group is able to breed to produce fertile offspring is the second part. This is exactly the wording or phrasing you need to use. So you need to be able to say that they're similar or they share characteristics and they produce fertile offspring. So let's unpick it to see why this is important. So we class tigers and lions as two separate species, although they are very, very similar in all of their physical features and characteristics because they are all of the same genus, because they both share the genus Panthera. So they've got all of the external and internal structures and features and probably very, very similar DNA. And they can successfully breed. That's how closely related they are. They can breed to produce offspring, which are known as ligers. However, ligers are infertile. So here's a picture of a liger. You can see it really shares those characteristics. It's got kind of a bit of a mane like a lion, but it's got that kind of tiger-shaped, sort of shorter face. So they can successfully breed only in captivity. They wouldn't do this naturally in the wild, but they have been made to breed in captivity. And the reason, therefore, we know that these two species are not the same species is because they produce ligers, but these ligers are infertile, which means they cannot go on to have their own offspring. This is obviously taken from the classification system. So we have the three domains. We have bacteria, we have archaea and eukaryota. So as I said, all the eukaryotic cell-based organisms are in the eukaryota. Then we have bacteria and we have archaea, which are their own groups, which is two different groups of prokaryotic cells. Within the eukaryotes, we have three kingdoms. We have animals, fungi, plants, and protists, four kingdoms. And then archaea have a kingdom of their own and bacteria have a kingdom of their own. So this is the 3-domain, 6-kingdom classification system. This is the modern classification system. Okay, so 3 domains and 6 kingdoms. Previously, we just used to have 5 kingdoms. So we used to have animals, plants, fungi, protists, and prokaryotes. And the prokaryotes group has now been split into bacteria and archaea to give those 6 kingdoms, and also we have added the group domain. This extra level, so the domain and the addition or the splitting of the prokaryotes into bacteria and archaea, was done by Carl Woese in the 1970s. This discovery was made and the decision made to change it because of advances in biochemistry technology. So we were able to look at the actual inside workings of the bacteria and the archaea. Also the electron microscope, which allowed us to identify and see inside structures of the bacteria and the archaea, enough to be able to say that they were different enough to be separate groups, which is what led to the creation of the 2 separate domains. So the bacteria domain and the bacteria kingdom contain what we call the true bacteria. So they have all the classic features of what we call the true bacteria groups. The archaea are known as the primitive bacterial group, and they include a lot of bacterial species that are extremophiles. So they have this ability to survive in really extreme habitats. So you'll see some of the names here. So like therm, that means it can survive in normally high temperatures. There are some that survive in really cold temperatures as well. And things like halophiles, these have a really high tolerance for salt. So there's lots of different sort of slightly strange bacteria in the archaea group. And they're considerably different in their biochemistry from bacteria, which is why they are able to be separate. The development of classification systems. So initially there were only 2 kingdoms, plants and animals. Both of them contained both the multicellular organisms, obviously plants or animals, but also single-celled organisms that shared characteristics with either plants or animals in their cells. So things with chloroplasts would have gone into plants, for example. This left an issue though, because microorganisms like Euglena, which has a flagellum and can move around, but has chloroplasts, doesn't really fit into either plants or animals quite neatly. And fungi as well are examples of ones that didn't fit neatly into plants or animals because they have a cell wall like plants, but they don't photosynthesize, so they didn't really fit. The development of electron microscopes allowed us to see more of these details inside these cells, which is why we realized they didn't fit into those groups. So then we found that these differences meant that we could split everything up into 5 kingdoms: animals, plants, fungi, protists, and then prokaryotes. Carl Woese was the scientist that used ribosomal RNA analysis to separate the prokaryotes to get our final classification system that we have today. So he found that bacteria have a different cell membrane structure. They use a different type of RNA polymerase enzyme. They don't have any histones, which are the proteins associated with the DNA molecule, and they have different ways of doing DNA replication and RNA synthesis compared to eukaryotic cells and also archaea. So he saw that actually this group of prokaryotes that included the bacteria and the archaea needed to be split because the archaea were much more like eukaryotes with some of their characteristics, and there's a group that were all of these bacterial organisms that had all of these differences that he found through using electron microscopes, ribosomal RNA analysis. Diagnostic features of the 5 kingdoms. So obviously we've spoken about the fact that there's now maybe 6 kingdoms, but we need to be able to look at the main 5 kingdom groups and to be able to say what the features of those groups and individuals of the groups are so that we can then identify organisms. So Animalia, anything that's in this animal group, must have eukaryotic cells. By that we mean obviously that they contain a nucleus. They must also be multicellular, and they're usually able to move around. They are heterotrophic, so they ingest other organisms and break down the large molecules and then absorb those small molecules they are then able to make new molecules with. The plants, they are also eukaryotic cells. They are also multicellular. They have a cellulose cell wall around their cells. They contain the pigment chlorophyll, and they are autotrophic. This means that they absorb small molecules and use them to build their larger molecules. The fungi, they also have eukaryotic cells. Their cell walls are made of chitin. They can be multinucleated, so that means they can have more than one nucleus within their cytoplasm. They can be single-celled or they can be multicellular, which means they create hyphae, which form a mycelium. Their form of nutrition is saprophytic, so they feed on dead organisms or dead organic matter or waste, and so they obviously help through part of that decay process. The Protoctista, or sometimes known as protists, they have eukaryotic cells as well. They are mostly single-celled, but there are some things like algae here, which is obviously a type of seaweed, can be. They are obviously multicellular. And there's a wide variety of forms in this group because it's basically the group where everything that's not an animal, a plant, or a fungi, or a prokaryote is put. Okay, so it's all the stuff that doesn't fit into those specific niche groups. So they have a lot of plant and animal-like features. They tend to be free-living, so living out as an individual, but they can be parasitic, so they can live inside a host. They can be autotrophic or heterotrophic, including things like they can carry out photosynthesis, obviously algae can. They can ingest prey, so actually engulf and digest prey, or they can secrete extracellular enzymes and then absorb nutrients that way, which is very similar to how fungi work. Prokaryotes. So to be in this group of prokaryotes, the cells must have no nucleus. They must have circular DNA, so a loop of DNA, not linear. Their DNA is not associated with the proteins that we know as histones. They don't have any internal organelles, such as mitochondria. They have smaller 70S ribosomes. Their cells are much smaller than eukaryotes. And they can be free-living, but some of them are parasitic. And we know, obviously, that some of them are pathogenic, so they live inside organisms and cause disease. So remember that this kingdom has now been split into 2 kingdoms, the archaea and bacteria, in the modern classification system. And this is because the archaea are actually more similar to eukaryotes than they are to bacteria. They have similar RNA polymerase enzyme. similar methods of replicating DNA and RNA, and they do produce some proteins that seem to bind to or associate to their DNA. So remember we went through the differences that bacteria had, that Carl Woese found, which is what led to the separation of this group. But just remember that you have these two groups, these two kingdoms now, that were the prokaryotes. So to summarise, taxonomy is the theory and the practice of biological classification. Classification is where we organise organisms into groups. Historically, as we said, going back to like Linnaeus and his classification system, and even before that, we classified organisms using physical characteristics and maybe other observable characteristics that were quite limited in order to try and spot similarities and say, well, these should therefore be in the same group as each other because they're very similar. But there was limited scientific equipment and knowledge of things like genetics at the time when a lot of these classification systems were made. Now we know that organisms can share physically similar characteristics, but they may not have the same evolutionary origin. So they're not actually evolved from each other or related to each other. They can be found in completely different times or completely different places on Earth, but they can have evolved similar characteristics because they're adapted to the same environment, for example. So things like flight, things like wings. There's lots of various examples where we call it convergent evolution, where organisms have adapted to similar environments but evolved in different places, and so they're separate, they're not related to each other. But now we have the technology to be able to use genetic investigation and molecular techniques to actually discover which ones are genetically related to each other, which ones did evolve from each other, and use that to help us when we're doing classification. And we call this type of classification, where we look at evolutionary relationships, biogenetics. And we're going to look a little bit of that in more detail in the next lesson.
Ouch! This is why in some videos I'll explain scratches.